Welcome to Avantor's Harnessing the Power of 3D with Corning webinar. Couple things before we begin. Just wanted to let you know that the recording of this will be recorded, and you will get a link to the recording after our session. We will also have a question and answer period at the end of this session. And we ask that you put in your questions in the question box at the top right-hand corner. And without further ado, I want to pass it over to Whitney Wilson from Corning to talk a little bit about the power of 3D.
Hi, everyone. Thank you for joining today's webinar. My name is Whitney Wilson, and I'm a field application scientist here at Corning Life Sciences. I've been with the company for a little over 3 years now, and prior to that, I directed the stem cell core at UC Davis for 12 years, where I worked in the lab with many different 3D models, including cerebral and cortical organoids. So today we will be discussing how you can harness the power of 3D cell culture utilizing Corning's technologies. So for those of you who are a little less familiar with our company, Corning is a material science company that's been innovating in the life sciences for over 100 years now. One of our very first products was our Pyrex glassware, which I'm sure you're all familiar with in one way or another, whether it's in the kitchen or in the lab. But what you may not know about Pyrex is that it actually helped to enable the early production and distribution of world-changing medicines such as penicillin and the polio vaccine. So we have been creating material science solutions for scientists throughout the last 100 years, and we continue to evolve our life sciences portfolio to meet the needs of a changing world and to ensure that our technologies keep up with scientific discovery. So at Corning, our products come from both internal innovation and acquisition and really cover the breadth of the scientific field, from the research laboratory to vaccine production and biomanufacturing to cell and gene therapy, cancer diagnostics and treatments, as well as final drug packaging with our pharmaceutical vials. So before we get into our 3D technologies, I'd like to start with an introduction to 3D cell culture to make sure that we all have the same background. So what do we mean when we say 2D versus 3D cell culture? In 2D culture, we rely on a typically inert surface for cells to attach to and grow. The cells all grow within the same plane on that surface and experience things like contact inhibition and limited physical space in which they can expand. In 3D cell culture, we allow the cells to grow in the X, Y, and Z plane, generating the formation of complex 3D structures or cells that are spatially separated from one another. So why do we care about 3D cell culture? The simple answer is that 3D cell culture is far more biologically relevant than 2D culture. The cells in our bodies obviously do not grow on a hard plastic 2D surface. Instead, they grow in a complex 3D environment surrounded by extracellular matrix and many different cell types. So the image here on the left is of a lung tumor growing in its endogenous environment. And as you can see from its morphology, it's not flat, but instead spherical, surrounded by other tissues in VCM. And as you can see in the diagram on the right, cells in a complex 3D environment experience things like non-uniform exposure to oxygen, nutrients, and drugs, ECM to cell and paracrine signaling, as well as proliferation gradients and increased cell-to-cell interaction. And all of these important biological factors can be very difficult to recapitulate in a 2D cell culture environment. So here I'd like to emphasize the difference we see in cell morphology between 2D and 3D cell culture. In 2D, our growth substrate is rigid and typically inert, and we observe an altered cell shape compared to what we would see in the body, as well as a loss of cell polarity. We see that growth kinetics are altered, and the architecture of the cell is much less physiological, with cells only partially interacting with one another. Growth factor diffusion across the cell population is rapid in 2D, and importantly, we see differential gene expression compared to what we would see endogenously. In contrast, in 3D cell culture, our growth substrate mimics the natural tissue environment, and we observe more similar cell morphology as we would see in the body, as well as the establishment of cell polarity. The growth kinetics of the cells are much more in vivo-like, and the architecture of the cell environment is more physiological with close interactions between the cells and ECM. In 3D, we also generate biochemical gradients with growth factor diffusion, and most importantly, we see that we maintain gene expression patterns that resemble what we would see in vivo. Traditionally, 2D models have been selected as preclinical models for drug discovery and disease modeling because of their ease of use and high throughput and scalability. However, when more biological complexity is required, scientists have historically turned to animal models, which of course can be challenging and expensive to use. Spheroids and organoids are really marrying the benefits of these two models, but also provide a more human-relevant model, as they're typically generated with human cells. And in fact, in September of 2022, Congress passed the FDA Modernization Act 2.0, which essentially authorizes the use of certain alternatives, such as cell-based assays, to animal models, no longer requiring the use of animal testing. So since passing this legislation, we have seen an increased demand for 3D cell culture products as investigators turn to 3D assays in lieu of animal. So often the words spheroid and organoid are used interchangeably, but they are actually distinctly different things. Spheroids are made from a cell line or primary cells and generally only contain one or a few different cell types. They do generate cell and concentration gradients, but they have limited self-organization and polarity. They also typically do not require an ECM to grow. In contrast, organoids are generated from stem or progenitor cells and contain multiple organ-specific cell types. They self-organize and exhibit polarity and typically do require some sort of ECM to grow. Most importantly, organoids can recapitulate organ function. So spheroids and organoids can be used for many different applications, including cancer modeling to study disease, pathophysiology to better understand mutational signatures. They can also be used for drug discovery and high throughput and CRISPR screens, as well as personalized medicine to identify treatments that are a molecular and pharmacogenomic match for the patient. They are also used in conjunction with bioprinting to allow scientists to study cellular relationships that affect tissue functionality. So next I will discuss various Corning tools that can be used to generate 3D models. Corning has a long history of being a leader in the 3D cell culture solution space. One of our first 3D products is Matrigel, which I'm sure many of you are familiar with in one aspect or another. Another oldie but goodie are our Transwell permeable supports. Some of our newer products include our spheroid microplates, as well as our aplasia plates and Matrigel for organoids, in which we have determined the specific elastic modulus of the ECM. And then we have our solutions for working in 3D with higher throughput, such as our Matrigel Matrix 3D plates, our Matrigel bioprinter, and the Aplasia Flask. So I'll be going over many of these products in more detail in the coming slides. When we culture cells in 3D, essentially there are three different systems that we use. The first utilizes a biological scaffold, such as a hydrogel, to create a 3D environment in which the cells can expand. The second is a scaffold-free system in which we utilize ultra-low attachment coating to prevent cells from attaching to plastic so that they can naturally aggregate and grow in 3D clusters. And then the third system utilizes a mechanical scaffold in which we physically separate the cells with a permeable support to allow for 3D-like interaction. Let's start with discussing scaffold-free systems in more detail. Almost all of our scaffold-free technologies utilize our Corning ultra-low attachment surface coating. This is essentially a hydrogel coating that prevents cell attachment to plastic and in turn promotes self-aggregation of the cells in suspension. The ULA coating has no special storage requirements and can be used on automated platforms. The first product I'll talk about is our spheroid microplate, in which you can generate a single spheroid per well utilizing our unique teardrop round bottom geometry. In this plate, you can control the spheroid size by adjusting your seeding density and ensure that you have uniform sized spheroids across the various wells. The plate is designed with black walls so that you can culture your cells and then assay within the same plate. And it comes in 96, 384, and 1536 well formats. And it's been referenced in literature for spheroid generation for over 40 different cell types. To generate spheroids in the plate, it is important to seed with a single cell suspension and then centrifuge the plate to promote cell aggregation. Over the course of several days, the spheroids will form and can be cultured for extended periods prior to assay. The exact formation, size, and duration of multicellular spheroids is dependent on the cell type and environmental conditions such as media formulation, the presence of Matrigel or other ECMs, or the presence of other cell types in a co-culture system. Because the size of the multicellular spheroids is cell line dependent, it is recommended to perform optimization testing of cell seeding conditions when working with a new cell line in the spheroid microplate. Another great product we have for spheroid generation is our Alplasia microcavity plate. These plates essentially have small microcavities within individual wells that allow the end user to bulk generate spheroids to increase assay signal or produce bulk RNA and protein. The plates are black walled to reduce crosstalk between the wells, and the 96 and 384 well formats are automation compatible. The Alplasia plates come in two formats, round bottom or square bottom. The round bottom plates have a 500 micron microcavity diameter to generate larger spheroids, and the square bottom plates have a microcavity diameter of 200 micron for slightly smaller spheroids. These plates are ideal for high content imaging. An important difference between the two is that the round bottom plates come with the ULA treatment, while the square bottom plates are plasma treated for self-coating with polyhema to prevent cell attachment. Both are available in 6, 24, and 96 well formats, but only the square option is available in 384. The chart on the right indicates how many microcavities there are per well in each format, which directly translates to how many spheroids you could generate in each well. If you are in need of producing even more spheroids in bulk, we have our Alplasia 12K microcavity flask in which you can generate about 12,000 spheroids. This flask is made with our ULA-coated optically clear gas permeable polystyrene film, which is very similar to our Hyper technology. And what this means is that you can culture spheroids in the microcavities for over 2 weeks with optimized gas exchange, since this is occurring directly at the point of spheroid location in the plate, instead of gas transfer through the air-liquid interface. The vessel has a liquid diverter at the front of the flask that allows for media exchange without dislodging the spheroids from the microcavity. It's very useful for bulk RNA and protein isolation, and we have also seen a number of customers using it for extracellular vesicle production in 3D as well. So similar to the 12K flask, we recently launched our 12K open well plate, in which you can remove the lid of the plate for robotic selection of individual spheroids. This plate has media exchange ports for media removal and addition without spheroid disruption, and is made of the same gas permeable film that the flask is for extended spheroid culture. If media volume is a consideration for you, this plate may also be a better option than the flask, as it utilizes about half of the media that the flask does, with recommended volume of 20 to 30 mils versus 25 to 50 mils in the flask. As you can see from the different microcavity shapes and designs across the Alplasia portfolio, investigators have the flexibility to generate spheroids of several different maximum sizes. It is very important to consider the microcavity size of the plate when determining which plate you will want to work with. So which plate should you use? If you need to keep your spheroids separated for your assay, the spheroid microplate will be the best option for you. If you need to increase your assay signal by producing identical spheroids in bulk, the Alplasia microcavity plates are a great choice. But if you need to produce identical spheroids at scale, the Alplasia 12K flask will be your best option. And if you need to access those different spheroids in bulk, the 12K open well plate will be your best product. One of the common issues that arises when generating spheroids is the formation of satellites. This can be mitigated by ensuring that you are seeding with a single cell suspension. And this can be optimized by increasing exposure to lifting reagents such as Accutase, as well as by utilizing a cell strainer prior to cell seeding. If you are needing to move your spheroids from one plate to another vessel or mix them in suspension, we offer wide bore tips that allow you to do this without disrupting the integrity of the spheroid. Our P200 tip has a bore size of 800 micron, and the P1000 is 1600 micron for gently moving larger organoid structures. And if you'd like to automate the counting of your spheroids or organoids, we have our Corning Cell Counter with an organoid counting software upgrade. The Cell Counter is a low-cost cell counting tool for mammalian cells and utilizes a standard human cytometer for cell counting, so it doesn't require any consumables. In addition to providing counts per milliliter, it will provide you with a surface area data for your organoids as well. And lastly, larger 3D structures can be more difficult to image. Our Corning 3D Clear is a tissue clearing reagent that promotes rapid tissue clearing for a variety of imaging and fluorescent applications. The top right, you can see some confocal microscopy images of non-cleared spheroids. And in the presence of just PBS, substantially less cells are identified. But once cleared with our solvent-based methodology, we can see the entirety of the spheroid without tissue shrinkage or morphology changes. So now we will move to the second 3D system we will discuss today, biological scaffolds. Biological hydrogels have long been considered the gold standard in 3D cell culture because they allow us to recapitulate the ECM environment, which in turn allows for cell polarity to form. With hydrogels, you have the flexibility in how you utilize them structurally, but they have been historically difficult to work with in high throughput due to temperature considerations. We've developed some tools to help with that I will go over in the coming slides. There are essentially four main ways that we culture cells on or in ECM. The first method on the left is a 2D method and is probably the most familiar to us all. Here we culture the cells on top of a thin coat of ECM, such as these iPSCs pictured here atop Matrigel on the left. The second method is a 3D method called embedding, in which we first mix the cells with the ECM and then plate this mixture into our vessel. This allows the cells to form 3D structures within the matrix that can be a difficult method to use for imaging applications, as you can see that the structures form in different focal planes. The 3D sandwich method helps to resolve this by controlling the plane in which the structures form. In this system, we first coat the vessel with a hydrogel and then plate the cells atop that hydrogel in a media mixed with a lower concentration of the hydrogel. And then finally, we have the 3D dome method, in which we embed the cells within the hydrogel and then plate the mixture in dome structures on a vessel. You have the option of keeping the domes attached to the plate or washing them off to culture them in a suspension platform. This is the method most often used for organoid culture. Corning has a very extensive portfolio of ECMs, ranging from animal-derived options, such as Matrigel, to our newly launched fully synthetic hydrogel called Synthogel. The substrate that you choose to use will be application dependent and will largely be determined by the biological noise that your assay will tolerate. Probably the most popular ECM hydrogel that we sell is our oldie but goodie Matrigel. It comes in various formulations and concentrations to be used for different applications. And I just want to highlight here that we do have an organoid-specific Matrigel in which we've determined the elastic modulus and confirmed that it is strong enough to support organoid growth over longer periods of time without necessitated re-embedding. Working with Matrigel requires some specific considerations around temperature, as it remains a liquid when cold but will start to gel at temperatures above 10 degrees Celsius. It is important to pre-chill all of your materials that will come in contact with Matrigel and work quickly to avoid polymerization. The Corning Cool Box is a great alternative to working on ice, especially when you are working inside of a biosafety cabinet. It is also helpful to use a positive displacement pipette and definitely avoid blowing out tips to prevent bubble formation. Each lot of Matrigel has its own specific protein concentration. It is very important to reference the certificate of analysis to calculate your dilutions in terms of protein concentration, not percentage. For some applications, you may need to remove the Matrigel for either staining, protein extraction, or passaging. Corning's cell recovery solution helps to depolymerize thick gels of Matrigel to free 3D structures. Essentially, you add chilled cell recovery solution to the Matrigel culture, and then break up the gel mechanically with a wide bore tip. Then you incubate the culture at 4 degrees Celsius and simply wash with cold PBN. We recently launched our new Synthegel hydrogel option, which I'd like to emphasize is not a Matrigel replacement and is a separate technology. Synthegel is essentially a synthetic peptide solution with a cross-linker that is stable at room temperature. You can tune the rigidity of the matrix by adjusting the peptide concentration, and we have various versions available that are optimized for 3D culture of iPSCs in both suspension and adherent embedded options, as well as a spheroid culture option. The Synthegel spheroid matrix can be particularly beneficial for generating nice-looking spheroids from cell lines that are either difficult to generate spheroids from or generate scraggly-looking spheroids in other ULA-based methodologies. The ECM is designed to be used as an embedded methodology and can keep the spheroids in suspension and from settling in culture. As you can see from the images on the bottom of this slide, by adjusting the peptide concentration, you can really change the size and morphology of the spheroids from the same exact cell line. One of the tools we have for increasing throughput when working with Matrigel is our Matrigel Matrix 3D plates. Essentially, these are plates that are pre-coated with Matrigel for seeding cells using the sandwich method. The major advantage of working with these plates versus self-coating is that we have built-in quality control to ensure consistent speed values well to maximize data collection speed when doing high-content imaging. For those of you doing organoid work, you may have experienced the incredible pain of generating hundreds of organoids manually. To help with this, we have our MatriBot bioprinter that will automate this process for you and can perform droplet printing as well as print basic structures. The system is about 1 cubic foot and can fit inside of a biosafety cabinet. It utilizes a cooled syringe print head and thus can print temperature-sensitive hydrogels such as Matrigel, among others like collagen alginate-based bioinks. One of the common annoyances that scientists have when culturing spheroids is the difficulty when changing media. To help with this, we have our Lambda Elite Max semi-automated liquid handler that can be programmed to perform consistent, gentle media exchange in our spheroid microplate. Okay, finally, we'll discuss the third 3D system in the Corning portfolio, our mechanical scaffold. So you're probably familiar with the term transwell, which is one of the many types of our Corning permeable supports. Essentially, in this system, we have an upper apical compartment that is physically separated from a lower basal compartment, which provides two separate surfaces within the same well where you can culture cells physically separated from one another. Permeable supports have infinite applications, and customers are always coming up with new ways to use them. But some of the primary uses are for simple tissue culture or co-culture, drug transport and toxicity assays, and migration and invasion assays. This lower image shows a newer application we developed in our applications lab, where we combine our spheroid microplate with our high-throughput screening transwell insert to generate a blood-brain barrier model to study cancer spheroids or brain organs. The table on the left highlights our many options available using different membrane materials, pore sizes, surface areas, formats, and coatings. There's a lot to choose from, and your application will determine which is the right option for you. You can always reach out to your local account manager, field application scientist, or our scientific support staff if you need help deciding which option to choose. So when picking a permeable support, the first consideration will be your membrane. If you plan to do imaging, you will likely need PET for its optically clear properties. If you need a higher pore density for diffusion studies, polycarbonate may be a better solution for you. However, if you require a membrane with strong chemical resistance, PTFE will be the best option. Since cells do not readily attach to PTFE, this membrane does come collagen coated. Now, if your cells require a coating normally to attach to a membrane, we have our BioCoat PET supports that come pre-coated with collagen, fibronectin, laminin, or matrix gel. The second thing you will need to determine is the pore size you will need. This will obviously depend on the assay you are performing. Do you want a larger pore so that cells can migrate through the membrane, or a smaller one so that they remain contained on one side or the other? The table on the right is a great assay-based selection guide to reference when making your decision. We also offer specialty membranes such as Fluoroblock, which is treated with a proprietary black dye for fluorescent imaging, as well as our previously mentioned BioCoat options. And then we have our high throughput screening Transwell available in both 24 and 96 well formats for running high throughput. Some common questions we get about permeable supports are around cell attachment, migration, and visualization. For attachment, it's important to consider if a coating substrate may be needed, and also your seeding density will need to be empirically determined as well. It can also be helpful to pre-incubate the permeable support to promote cell attachment. For migration, you'll need to make sure your pore size is larger than the cell type you are working with, and whether or not your ECM is too concentrated and blocking the pores. Also, is your attractant concentration sufficient to attract the cells to the other side of the membrane? And then for imaging, you need to make sure you are using a PET membrane and consider staining as an option as well. Finally, we'll discuss some specific 3D modeling examples. There are many different applications of 3D cell culture, including basic research, drug screening, biobanking, genetic profiling, cancer modeling, genetic engineering, and personalized medicine. In the coming slides, I'll highlight a few cancer models that utilize the three different 3D methodologies we discussed: scaffold-free, biological scaffolds, and mechanical scaffolds. In this first example, we utilize a scaffold-free methodology using our spheroid microplate in conjunction with Promega's Cell Titer Glow 3D viability assay for growth analysis and drug screening. We first generate spheroids from 4 different tumor cell lines and then expose the spheroids to doxorubicin to generate a dose-response curve. This is a great high-throughput method for tumor spheroid growth measurement in response to a specific drug of interest. In this second scaffold-free example, we perform CAR-T screening for tumor cytotoxicity, again using our spheroid microplate, and this time the DiscoverX killer cytotoxicity assay. Essentially, target killer cells are seeded to form tumor spheroids over the course of 48 hours, and then CAR-T cells are added, followed by the detection reagent 24 hours later to quantify tumor cytotoxicity using luminescence. And in effect, what we see is that the CAR-T cells effectively lyse the target cells after 24 hours. But what I'd like to highlight here is that when compared to 2D culture, the CAR-T cells are almost 40% less effective at lysing the target cells in the biologically relevant 3D models. This demonstrates the importance of utilizing 3D models instead of 2D models to more accurately predict clinical outcomes. This third example uses the scaffold-based sandwich method, utilizing our Corning Matrigel Matrix 3D plate. Here we seed the pre-coated Matrigel plate with pancreatic cancer cells to form 3D structures over the course of 48 hours. We then perform high-throughput cytotoxicity screening to target this specific cancer and determine the ideal dose for each drug candidate. The last 2 models that we'll discuss today utilize a mechanical scaffold. The first here is an immune oncology model pairing the spheroid microplate with the high-throughput screening Transwell permeable support. In this model, we are measuring the migration and cytotoxicity of natural killer cells. GFP-expressing A549 tumor spheroids are grown in a spheroid microplate for 24 hours, and we then add the high-throughput screening Transwell to the culture plate with the pre-labeled NK cells added to the apical side of the insert. The plates are then incubated together with a chemokine DDF1 alpha added to the basal side of the culture to assess the chemotactic response of the NK cells. At assay completion, live cells are quantified as a measurement of tumor cytotoxicity. In figures A and B, we observe that the ecadherin and GFP-labeled A549 tumor spheroid has been successfully infiltrated by the CD45 and cell tracker blue labeled NK cells. The NK cells have been able to migrate through the transwell pores and have demonstrated cytotoxicity. The last model we will discuss is an in vitro blood-brain barrier model. In this model, we again use the Corning spheroid microplate in conjunction with the high-throughput strain transwell permeable support in order to assess the ability of certain drugs to pass through the blood-brain barrier. We first established the blood-brain barrier by seeding PGP-expressing MDCK2 MDR1 cells on top of the high-throughput screening transwell. After 5 days in culture, we assessed the effectiveness of the blood-brain barrier model to ensure that tight cell-to-cell junctions have formed via immunofluorescence staining of ZO1 and occludin, as well as by measuring leakage of lucifer yellow from the apical to the basolateral compartment. Additionally, we look at PGP pump function to check for a higher efflux of the PGP pump substrate RH123 in the basolateral to apical direction. Separately, we generate a glioma sphere in the spheroid microplate by seeding LN229 cells and allow them to aggregate over 24 hours. On day 5, we combine the blood-brain barrier transwell model with the glioma spheres and expose the individual wells to a library of drugs for 2 hours. After incubation, we remove the transwell insert and reconfirm the blood-brain barrier integrity. We then culture the spheroids for an additional 2 days, after which we assay the spheroids with the CellTiter Glow 3D assay to assess for viability. And what we observe is that the addition of the blood-brain barrier significantly reduces the amount of drug hits we get by over a half. Without the blood-brain barrier, we get 27 hits, but with it, only 9. This assay again highlights how important biorelevant 3D models are when performing drug screening. Finally, I'd like to make sure you are aware of the many Corning resources available to you. In addition to local account managers and field application scientists such as myself, we also have a scientific support team available by email and phone that you can send specific application and technical questions to. They will typically get back to you within 48 hours with their suggestions. And then, of course, you can always reach out to our general customer service. And with that, I'd like to thank you for joining our webinar and answer any questions the audience may have. Thank you.
Thank you, Whitney. I see one question in the box here from Brianna, just asking about the clearing solution and wondering if with the clearing solution, can this be removed and then continue culturing beyond that?
No, this is for the termination of the culture.
Okay.
That's a good question.
Another one here is it difficult to keep the spheroids in individual microcavities in the aplasia vessel over time in culture? Do they easily get jarred out and aggregate and other spheroids to form like the Mickey Mouse structures?
Mm-hmm.
You know what I mean?
Yeah, I know that this is a common issue with other types of microcavity plates. The Corning plates are actually designed with deeper microcavity wells, so it really isn't an issue as much as with other platforms. You still, of course, need to be careful when transferring your plate to the microscope or for feeding, but generally it's not as big of an issue as with other types of plates.
Gotcha. Sticking with spheroids, is it difficult to have similarly sized spheroids in individual wells, even if you think your pipetting technique is good? Is there anything that might happen if people aren't finding uniform spheroids?
Yeah, so usually the most common culprit of not being able to generate uniform spheroids across either the spheroid microplate or even in aplasia is if you're not starting with a truly single cell suspension outright. So if you do have some clumps of cells, maybe you didn't allow your protease or enzyme to be exposed to your cells when you lifted them in the first place for long enough, then you'll have these kind of clumps and clusters, and then you won't be able to efficiently generate uniform size spheroids.
Thank you. And speaking about Synthogel a little, can that actually be used to embed organoids or just for spheroids?
That's a really good question. I get that question a lot. So the Synthogel product has really only been tested so far for spheroid culture. However, I do have several customers that are using it to differentiate iPSCs into specific organoid types in the system. We just don't have a lot of data around that yet, but we hope to soon.
Okay. And some other common customer questions is sometimes they have trouble making domes with Matrigel on tissue culture plates, even with the same lot of Matrigel. Do you have any tips to help make that process more consistent?
Yeah, so I am totally guilty of having done that myself in the past. Blame the Matrigel instead of myself for my inability to make really nice domes. But usually the biggest culprit, more than the Matrigel, is actually the plate that you are using to generate the domes on. So when we tissue culture treat plates, we add a charge to the plate to essentially make it more hydrophilic. And so when you use tissue culture treated plates, or even plates that have even a higher tissue culture treatment, such as CellBind, you actually it actually works against your process because it increases the wettability, and so will flatten the dome. So the trick to getting better, more kind of punctate or upright domes is to work with either non-tissue culture treated plates, or if all you have in the lab are tissue culture treated plates, a trick is you can put them in the incubator overnight to preheat them, and that will actually help to form more upright spheroids when you're doing that process.
That's a good suggestion. All right. And then lastly, maybe with staining of organoids embedded in Matrigel, that can be an issue. Is there any suggestions for improving the immunofluorescence signal?
Yeah, so when you have your organoids embedded in Matrigel, it can be a little bit more difficult to do end-stage staining. So there are a couple things you can do. One is you can try the cell release solution, where you essentially remove the Matrigel from the spheroid prior to fixing the spheroid. But the other thing that I actually learned from my good friend who's a field application scientist at Molecular Devices is that essentially when you stain spheroids, it just takes longer for the different antibodies or stains that you're using to penetrate into the spheroid. So really, if you just increase the exposure time, so like increase the exposure of your primary antibody from, I think typically in like 2D, we do it for like an hour or 2 hours or maybe overnight. But if you increase that exposure time, you will see better penetration of your antibody or stain into the spheroid.
Very good. Well, any more questions from the audience? Put in the question box, and I don't see anything else, but I'm gonna give it a second and then feel free to close it out.
All right. I see some folks are starting to drop already, so I will close this out. Thank you so much for attending, everyone, and I hope that this was helpful.